Working? Working? Yeah. Hello, everybody. I'm Dr. Gabriel Costa, postdoc at University of Liège. And as introduced by Krista and Renzo, I'm going to give some overview on what we have on the functional notation. We have been listening on the last days a lot about functional notation and how to improve it, and what is the contribution of BovReg to the functional annotation of the bovine genome. As Krista introduced, the BovReg consortium brings together leading labs in the EU, and one of the main goals of the BovReg is to improve our understanding of the cattle genome, providing a comprehensive map of functionally active elements in the bovine genome. We believe that it's critical to link genotype to phenotype, as we have been discussing in the last presentations and during the whole conference. Recent efforts from FAANG community and also from different projects have put together functional notation on the bovine genome, but most of the time limited by the number of tissues, number of breeds, and number of samples utilized. Here I'm showing some of the examples of the most recent papers published on the functional notation of bovine, and showing that we have a very limited number of tissues and samples and breeds represented on the whole functional notation of bovine. A recent paper in Nature Genetics has discussed that we are still missing diversity on epigenetic studies in humans. And in humans, we have plenty of assays applied on different tissues, on different populations. And we don't have the same scenario for the livestock animals, for bovine, for example. And what is the contribution of BovReg? We decided to do a comprehensive functional notation of the bovine genome, putting together animals collected on different conditions, different sexes, different environments, development stages, and different breeds. What is this data set that we have collected? From 6 different animals collected from different countries, we have 2 neonate animals from Belgium, which are hosting breed, 2 juvenile animals more or less 7 months old, which are Kinsella composite and are collected from Canada, and 2 sexually mature animals or adult animals collected from Germany, which are Chevrolet crossed with German Holstein F2. From these 6 animals, we collected at least 24 different tissue types from each one of those, comprising a total of 129 different tissue samples. On this 129, we applied mRNA or poly-A assay. We also applied total RNA, small RNA assay, cage experiment, attack-seq, RABS and methylation, as Kristen mentioned before, and ChIP-seq for the classical stone marks and CTCF, which would allow us to predict and annotate enhancers, promoters, and so on the bovine genome. It's important to mention that all the time that you see the symbol of ULiège, it means that all the samples were processed, all the assays were processed, produced by the same person on the same lab. So to avoid and reduce the bias that we have on the data generation. On the transcriptome atlas of BovReg, we have annotated about 48,000 genes, which is already way more when we compare with Ensemble and NCBI available annotations. We have annotated about 300,000 transcripts, and for part of those, we already have Ensemble biotypes assigned, but we also could assign CDS and start and stop codon for some of those, and we have a large catalog of non-coding transcripts. We have enhanced the annotation of non-coding transcripts using not only poly-A but also total RNA assay, which allow us to detect transcripts which are not polyadenylated. From the total content putting together mRNA or poly-A and total RNA assay, from the whole panel of 300,000 transcripts annotated, we identified at least 15,000 which are completely novel transcripts, doesn't have any kind of overlap on what we have on the existing annotations of Ensemble and NCBI. And it's what I'm highlighting here in red. By applying the small RNA assay, we have identified 1,265 microRNAs, which by number is already more from what we know from MeBase. Half of those were annotated as novel microRNAs, no overlaps, and interestingly, most parts of them overlaps a specific transposon family, DNA marina transposon, which is not active in bovine anymore. It's a DNA transposon, and we don't know if it's a biological meaning or if it's a technical issue because of the size of the mature sequence of the microRNAs. But it's an interesting observation that we can try to pursue. On the other half of the microRNAs, which are already annotated, indeed most of them are annotated in bovine, but we still have a few which are annotated in different species, but not in bovine. And it's one of the things that we are adding on the transcriptome annotation. Here I have one example of a microRNA which is annotated in different species and is highly expressed on skeletal muscle longissimus dorsi, only on the neonate animals, not on the juvenile or adult animals. And that's why we believe that this microRNA was not annotated before in bovine, because most of the studies are using adult animal instead of the different development stages. Here on this graph, we have the expression all over the 129 tissue samples normalized by CPM. And this microRNA is located within a very well-known imprinted domain, which we already have the annotation of plenty of other known microRNAs, but this one, we are adding the annotation on the BovReg consortium. We also did cage experiment to refine the annotation of TSS in bovine. This paper is already published. It was led by UK, by Emily Clark team. And on this analysis, we have refined a 1,000 TSS annotation in bovine and 2,300 more or less TSS enhancers annotated in bovine. When we look at the genomic features comparing with ensemble annotation, we see that 22% of the TSS are annotated on intergenic regions. We believe that by comparing with the annotation from BovReg, now improving the number of genes, we can have a different scenario. It's also interesting to show here in this graph, we have the number of TSS annotated on the different species, and in gray, is the ones that are specific for each species, in yellow, the mammalian one, and in orange, the avian mammalian homologs. We can see that in bovine, we have 4,700 TSS, which are Bos-Taros specific. On the regulatory regions, we have applied ATAC-seq and ChIP-seq to the best of our knowledge for the first time for this number of samples on different stages and conditions. We have identified thousands of peaks for ATAC-seq on open chromatin regions from the different stone marks, as I'm highlighting here the possible association. We need to combine them to have a better annotation, but we use the classical ones, and also CTCF binding sites. I put my face on A3K27 methylation 3 because this one is associated with polycomb repressed and is silencing mechanism. It's one of the tricky stone mark that we have to interrogate on ChIP-seq. We generally have a very high background signal, and it's quite difficult to work with this one. And that's why we don't have the pretty consistent number of peaks across the different samples. But for the other ones, we can see that we have a consistent results for peak calling. Putting together all these peaks that I mentioned before, trying to predict and annotate the different status of the chromatin, we use the chromatin H&M software to have the estimation of the probabilities of signal for each one of the stone marks interrogated. On this picture in blue, it's exactly the emission probabilities. When we have dark blue, you have a high emission probability, and we could assign at least 14 different chromatin states, putting together all the ATAC-seq and ChIP-seq data from BovReg. And here we have also the percentage of the genome that's covered by the annotation. We could annotation at least 25% of the cattle genome by combining ATAC-seq and ChIP-seq results on the 129 samples. I want to highlight that inspecting some of the active enhancers that we annotated, we could identify liver-specific here in this example, but we could identify tissue-specific regulatory elements, in this case enhancer, which only present on the neonate animal which has 2 weeks old. This regulatory element was not detected on the juvenile and adult animal. It's one more contribution from BovReg on the comprehensive functional annotation. We also use the state 13, which is the active TSS promoter region, annotated by ATAC-seq and ChIP-seq, to validate the TSS that we annotated from the transcriptome map. By doing so, by checking the overlap, we have 79% of the TSS detected by RNA-seq that overlaps with the same stage from attack and ChIP-seq, corroborating that indeed we have a good quality annotation on the transcriptome map that have been validated by the prediction of the chromatin states with attack and ChIP-seq. We also compare our data with the most recent paper from the team of UC Davis from FAANG US, where they did ChIP-seq in 8 different tissues. We took exactly the same tissues and we compare our chromatin states predicted with the same states that they predicted. And in red, we have the percentage of active enhancers that we could recover, the ones that they red annotate. We can see that we could recover most of them using exactly the same tissues, and as we used way more tissues, we could detect novel ones, as I'm showing in these 2 pictures below for liver and spleen. We have each bar, it's each one of the 6 animals of BovReg. In blue is the number of active enhancers that are detected only by BovReg, and in orange the ones that are detected by both researchers or both studies. We can see that by adding diversity in our data, we could recover most of the known ones, but also add new ones detected on BovReg consortium. It's also important to mention that we have identified mobile genetic element polymorphic insertions on cattle genome, and for the first time we deployed about 700 probes that are unfixed on the EuroGenomics array. These probes are already available for the public and could be used to genotype the different breeds and see how these insertions are segregating on the different breeds. Here, overlapping the position of the 700 mobile genetic elements with the chromatin states predicted by BovReg, we have identified co-localized mobile genetic element insertions with regulatory regions. Here is an example of liver-specific enhancers which co-localize with mobile genetic element insertion, specifically a ERV21, which is a family that's still active in cattle. And we see here, citing one of recent papers on pangenome, we believe that transposable elements will be increasingly being detected as driving forces on the phenotypic diversity in bovine. So this information is already public, available for the public, and can be used to interrogate on different phenotypes. As perspectives and take-home message, we have generated a large catalog of genomic features that will now be validated and be useful for the different researchers and the community. I'm listing here some of the consensus peaks, as Renzo mentioned, and some of the main results that we have from transcriptome map, regulatory regions, and variations, as well as the EQTL that Krista mentioned before for different tissues and breeds, and also mentioned by Renzo, that are currently being used on genomic prediction and see if we have an effect using the different genome features of BovReg. Most of the interesting results will be communicated and public by the end of the consortium in February, so stay updated and follow us to see some of the interesting achievement. And I would like to thank all the partners and open for questions. Thank you very much.